So I'm really excited to talk today about my work on the effect of planting density on carrot seedling establishment and canopy growth. Just a general outline, I'm going to give you a little bit of background on why top size is important in carrots. A lot of the emphasis is on the root, but I work on the above ground portion. The objectives of the study, a little bit about the experimental design and the model I used to look at the data, and then I'm going to focus on the effects on germination, canopy height, and some harvest measurements of quality, and discuss some future work. So carrots are grown on a lot of organic farms, and about 14% of overall U.S. carrot production is on organic land. But weed management is a huge challenge, and this is largely due to the growth habit of carrot. They take a long time to germinate, around 2 to 3 weeks. They're slow to establish and also very slow to develop canopy structure. So the idea is to find varieties that have a competitive growth habit and may be better able to compete for light with weeds. So the objectives of this study were to compare 8 diverse breeding stocks of small and large tops at multiple organic locations, and also to evaluate the effect of planting density on canopy growth, source sink allocation between the shoot and the root, and overall yield. So I used a split plot design. I had 3 locations at the whole plot level. These were then subdivided into 3 planting densities, low, medium, and high, which on average were about 16, 35, and 44 plants per meter. And there were 3 reps of this at each location. And this was further subdivided into 8 genotypes, the sub-plot level. And there are 4 small top genotypes, which are coded A through D through the presentation, and 4 large tops, E through H. And these were identified based on prior field observations on conventional land and at the end of the season. So just a little bit about the locations. We've heard a lot about the UW-Madison Research Station today, so that's included. TP Produce in Evansville, which is managed by Steve Pincus and Beth Kazmar, and Standard Process in Palmyra, managed by Christine Mason, and they actually produce organic supplements, kind of interesting. And they were all certified by the Midwest Organic Services Association. Some of the measurements I took included germination percentage, height and width measurements of the canopy, 3 measurements per plot weekly, and the number of marketable roots, so just, which I just defined as above a certain size threshold, so anything that was clearly immature was not included in that measurement. And root and top weights, so fresh and dry weights. I just want to point out some interesting aspects of the raw data. This is looking at days after planting and corresponding plant height for the 3 different locations and the 8 genotypes. Again, A through D are small and E through H are large. But the key points I want to make with this are there's a lot of variation among locations, which we've been discussing a lot today. And also that you can start to detect genotypic differences about 7 weeks after planting. And you start to see sort of a clear separation between large and small, and this is consistent across locations. The model I used is a mixed effects model. I fit it in LME4 and R. It's a very nice package. And the main effects were location, planting density, and genotype, and all subsequent 2nd and 3rd order interactions. And rep was included as a random effect, rep by location to estimate the whole plot error term, and rep by location by density for the subplot error term. So the ANOVA table, it can be broken down then into 3 parts. The whole plot, which measures the effective location. The subplot, which measures density and location by density interaction, and the sub-subplot, which looks at genotype, genotype by location, density by location, and the 3rd order interaction. And this just summarizes the significance of each factor for different measurements, but I'm going to focus specifically on the effect of planting density, which was significant for all the traits I measured except germination, and the genotype by location interaction, which was significant at the 0.1 level for all factors. Looking at germination, this is a really depressing graph. I had less than 50% germination in all of my locations for all of my genotypes. I had originally aimed to plant thick and thin down to my target densities, but I didn't actually reach the targets. So these are the 8 genotypes, percent germination, and then the 3 locations. And there's also a significant genotype by location interaction, with some varieties very stable and some showing a lot of variation among locations. Looking at canopy height, I'm showing data from early and late in the season. So 3 weeks after planting, there's this graph shows different planting densities, so low, medium, and high plant height. And then these blue dots are the estimated means from the model. The blue bars are the 95% confidence intervals of those means, and these red arrows are the result of multiple comparisons testing with the 2 key corrections. So basically, if the red arrows are non-overlapping, it's significantly different at the 0.05 level. So here there is a significant effect on plant height, with low planting density significantly lower than medium and high planting densities. And at the end of the season, it was really interesting. There's a significant density by location interaction. So there was no effect of planting density late in the season on plant height for standard process and teepee, but in West Madison there's a very significant difference between low and medium, and also medium and high. So it doesn't affect overall plant height at the end of the season. And if you look at this in another way, again, A through D is small genotypes, E through H is large, and at 3 weeks after planting, there's really no sort of separation between small and large tops, and also there's a lot of differences among locations. But if you look at 12 weeks after planting, not only are there fewer differences among the locations, but there's a much more clear separation between large and small tops, which is consistent across locations. And the best part of this graph that I thought was really fascinating is genotype B had very tall and rapid initial growth, but is still a small top at the end of the season, so has very rapid initial growth that diminishes over time, which is sort of ideal in that you can outcompete weeds early in the season until canopy closure, but avoid the accumulation of foliar diseases by having too much above ground tissue. So just in summary, higher planting density increases plant height during early growth, but had less effect at the end of the season. And although the genotype by location interaction was significant, the genotypes still match our expectations. They're either small or large consistently. Next, I'm going to focus on some harvest measurements. So looking at shoot biomass for single plants. Again, different planting densities and then the corresponding shoot biomass. Plants at higher density had significantly lower shoot biomass per plant than those at higher density. And the genotype by location interaction, there was a lot more variability among large tops by location for shoot biomass than for small tops. Looking at root biomass, the results were comparable, very similar, and it should be no surprise that there was no significant effect of planting density on the shoot weight to root weight ratio, which is something I was very interested in and expected to find a difference in, which I'll discuss a little later. Looking at the number of marketable roots, which is again just roots above a certain size threshold, higher planting densities had a lower percentage of marketable roots, but the important thing is the total number of marketable roots, which was much higher at higher planting densities. So maybe better to plant thick and increase your yield. Just in summary, although there were a lot of increases in plant canopy height at higher densities, the shoot and root biomass per plant was lower. There was no significant effect of planting density on biomass partitioning between the shoot and the root, and small top genotypes have less variation among locations than large top genotypes. And I had a very interesting conversation with Steve Pincus at TP Produce, and he mentioned that he will actually plant thick and extend the growing season so the roots reach a comparable size, but he also said they were higher quality, which I thought was interesting. In conclusion, there's a competitive growth response to planting density that was most substantial during early growth. There's a reduction of plant weights at higher density that's consistent with findings in a greenhouse study by Lee et al. And planting density did not significantly affect the ratio of shoot weight to root weight, which was observed in a study by Hull et al. That reported increasing ratio at higher planting densities of carrot. And I wanted to point out that might be due to that I did not include undersized or immature roots in my estimate of dry matter, and if I had, as you can see in this picture, the shoot to root ratio would be much higher and would skew that in the direction reported by Hull et al. Just from a breeding standpoint, selection may be most effective 7 or more weeks after growth or after planting, when genotypes can start to be distinguished. Genotype by location was significant, but for all traits that I measured, but I would argue that you can still consistently distinguish small and large top genotypes, and it of course emphasizes the need for testing in multiple environments. This summer, I want to repeat this. I would really love to hear suggestions from anyone or thoughts. I'm going to aim for thicker planting densities, additional genotypes, as well as a wild accession. There are a lot of reports that wild carrot actually does not have a competitive growth response. And I also want to incorporate a mid-season dry matter estimate. I just want to acknowledge Phil, my advisor, Pam and Shelby, who helped me take measurements, Erin Silva and her lab, Anders, Chloe, Eric, and Anne specifically, who helped me take measurements, plant, do all the weeding, which was substantial, Steve Pincus and Beth Kazmar at TP, Christine Mason at Standard Process for allowing me to use their land, and Cole Cook at Stats Consulting, who has been very helpful in analyzing this data. And of course, funding sources from USDA OREI and the Roger Globon Student Travel Fund.